Yeah, dear friends and colleagues, on behalf of my co-chair, Professor from welcome to this new USDR. This that is called Fresh Bacon for you today, and we will directly start with the first one, which is by Catherine Pei-Lu. She was in Elaine Fuchs' lab previously. She's still at New York University, has her own lab, and very recently published a very nice immunity paper. It's a very early start for you this day. We are even more grateful that you're with us, Catherine. Please, the stage is yours. Give us your presentation.
Let me share my, can you hear me well? Can you hear me well?
Yes, we can hear you.
Yes.
Okay. Thank you so much for the invitation. It's a true honor to have the opportunity to present our work on hidradenitis suppurativa as part of the ESDR Kitchen series. Hidradenitis suppurativa is a chronic inflammatory skin disorder that primarily affects human apocrine gland-bearing skin, such as axilla and groin. Although HS is linked to various autoimmune and metabolic conditions, we still don't fully understand its underlying etiology and pathogenic mechanisms. Clinically, patients present with painful nodules, abscesses, severe ulcerations. A hallmark feature of HS is the development of an epithelialized tunnel that extends deep into the dermis with massive immune cell infiltration and aggregation. Management remains extremely challenging, while therapy is the first FDA-approved treatment for HS. More than half of the patients fail to respond, and many continue to experience disease progression and recurrence over time. In order to develop more effective treatment, we need to acquire deeper understanding of the underlying nature of HS. To analyze immune cell composition within HS lesion, we perform multiplex immunoimaging. Wen is an excellent dermatopathologist from Taiwan. She found that within the dense immune infiltration, some of them actually have very well organized T cell zone, and in the middle, occasionally using single-cell RNA-seq, several other groups had also nicely discovered immune in the HS. However, we focus on proliferating cells and identify that majority of them are plasmablast. Our multiplex imaging confirmed a significant number of proliferating plasma cells located adjacent to HS tunnels. Furthermore, we perform single-cell B-cell receptor sequencing for patients' blood, perilesion, and lesional skin samples. Clonal sites are color-coded, as red indicate larger clones, while gray represents single cells. As you can see from the bar graph, we found that majority of the clonal expansion occurs in lesional skin rather than perilesional skin or blood. Technician, she successfully performed clonal tracking by utilizing a unique barcode of each cell within the same patient. Here we show that we think that in the lesional skin, larger clones are represented by red ribbons, and smaller clones are represented by orange, and single cells by gray. And then with cells from larger clones in the lesional skin are detected in the blood, as shown in these few red stars. And then the smaller clones in the skin are also present in the blood as single cells, as shown in the orange stars. Together, this result demonstrates that plasma cell clonal expansion occurs in the lesional skin and may circulate systematically in patient's body. Using heavy chain and light chain sequence from the most expanded plasma cell clone, we generate antibody in vitro, conjugated them with fluorophore, and perform tissue staining. We found that these patient-derived antibodies are reactive to a specific layer of keratinocyte in the lesional epidermis of the same patient or of different patient. Furthermore, we performed direct immunofluorescence to detect endogenous IgG and found a significant IgG deposition in the same layer of the tunnel epithelium. Using indirect IF with patient serum, we see a strong reactivity to the same layer as well, suggesting the presence of potential autoantigen at the site. Indeed, together we observe significant colocalization of endogenous IgG and serum antibody, as shown in yellow, in the epidermis, but not in dermis, suggesting that this layer of keratinocytes may be the site of immune complex formation. Next, we would like to know whether and how skin microenvironment may contribute to TLS formation. From single-cell RNA-seq data, we found that 2 fibroblast population express high level of CXL13 and CCL19, which may actively recruit lymphocytes to the skin. Additionally, we found that these fibroblasts express signature genes resembling the stromal cells in secondary lymphoid organs. We then hypothesized that skin fibroblasts may upregulate the expression of CXL13 and CCL19 upon receiving cytokines from B cell, T cell, and myeloid through various receptors. We then developed fibroblast culture from explant tissue and test each one of the cytokine to examine which one may effectively upregulate the expression of CXL13 and CCL19. We found that TNF alpha and LT beta stimulation consistently upregulate both chemokine in lesional fibroblasts. Jay is a talented postdoc in the lab with a bioengineering PhD. She utilized the microfluidic device to develop a TLS on the chip system. This device consists of 2 compartments. Fibroblasts are seated in the outer ring with metrogel, and T and B cells are seated in the inner circle. When normal fibroblasts are cultured with lymphocytes for 3 days, the lymphocytes remain scattered. In contrast, HS fibroblasts induce significant lymphocyte aggregation. We were able to label the T cell and B cell with different color and perform live imaging to analyze their dynamics over time. Treatment with additional CXL13, CCL19 further promotes formation of larger aggregates. Last, we show that anti-TNF antibody can effectively block the initiation of lymphocyte aggregation, but not once the aggregates have already formed. This result potentially explains why TNF blockade may be less effective in severe HS cases with extensive immune cell aggregation. In summary, we demonstrate that fibroblasts orchestrate the immune signaling circuits within the HS lesion, and that leads to lymphoid aggregation, formation of tertiary lymphoid structure, and as a result, plasma cell differentiation, proliferation, clonal expansion produce antibody that targets lesional epidermis. And this may contribute to downstream pathogenic events, which awaits further investigation. I would like to thank everyone in my lab, collaborators, and funding sources. We have postdoc position available. We welcome talented student and postdoc to join us. Thank you very much for your attention.
Thank you very much, Catherine, for this excellent presentation. Now we are going to switch directly to the next presentation by Tommaso Vicanolo. So Tommaso is a PhD student in Andres Hidalgo Labs in the Spanish National Center for Cardiovascular Research in Madrid, Spain. His research focuses on the role of innate immune cells in maintaining tissue homeostasis, with a particular emphasis on those cells to adapt local cues and to contribute to reinforcing the skin barrier. So now, Tommaso is going to present now this outstanding work published in Nature a few weeks ago. So please, Tommaso, the stage is yours.
Thank you. So thank you for the introduction and for the opportunity to present my recently published paper, which is entitled Matrix-Producing Neutrophil, Populate and Shield the Skin. So the organisms are protected from the outside environment by a series of physical barriers, among which the skin protects against mechanical injuries and prevent the entry of pathogens. However, when pathogens overcome these barriers, innate immune cells are rapidly recruited to the sites of infection, and they use the arsenal to eradicate infection. Neutrophils are a pivotal type of cells in this type of process. So neutrophils are the most abundant white blood cells in circulation, and they've always been described as inflammatory cells. So when you see a neutrophil in a tissue, it means there is inflammation. But in recent years, in our labs, we actually found out that neutrophils are present in almost every tissue, even if in homeostatic conditions, so without inflammation. So the question is, what are neutrophils doing there where they're not killing any pathogens? Could it be that neutrophils have evolved complementary strategies for organismal defense? We got actually inspired by a beautiful work from 2020 in which they found out that structural cells, specifically endothelial, epithelial cells, and fibroblasts, have encoded an immune potential in the epigenome already under tissue homeostasis, which it was triggered in response to systemic viral infection. So basically, they showed that structural cells can also act or have some immunological aspects to it. So we wonder whether immune cells can also engage into structural programs. So we performed RNA-seq from neutrophil deriving from hematopoietic tissues such as blood, bone marrow, and spleen, and compared them with neutrophil deriving from barrier tissues such as lung, gut, and skin. As you can see here, the neutrophils that derive from barrier tissue have an extracellular matrix signature featuring various collagens isoforms, and as well other major extracellular matrix transcripts such as elastin, fibronectin, laminin, but also enzyme related to the remodeling of the matrix, such as metalloproteases and fiber-forming enzyme. So we also validated this finding at the protein level. We used confocal microscopy, and we saw that in the skin under homeostatic condition, neutrophils are present, and the majority of neutrophils presented collagen 3A1, a prototypical matrix protein, inside of the cell bodies. Moreover, we also wonder where neutrophils were located in the skin. And as you can see here, using multiphoton 3D microscopy, we found out that neutrophils preferentially located in the subepidermal region, so right under the epidermis. Since we found out the neutrophil have the RNA, have the protein, the collagen protein, we took advantage of a really nice mouse model, which is called CNA35. CNA35 is a collagen binding protein from bacterial origin. And in this mouse model, the CNA35 is conjugated to mCherry, so it's visible by immunofluorescence, and it binds collagen trimers. In this way, we can identify which cells produce what type of collagen. So we crossed this mouse with a neutrophil-specific mouse model using the LYCG3 and a fibroblast mouse model using the DPT-CRT2 to visualize the collagens produced by neutrophil versus the collagen produced by fibroblasts. So, of course, as expected, fibroblasts are the main producers of collagens, but we saw that neutrophils also produce collagen, and the localization of these collagen fibers from these two types of cells is actually different. We saw that fibroblasts deposit collagen trimers more in the epidermis, while neutrophils deposit collagens closer to the epidermis, which aligns with the specific localization of neutrophil close to this area. So since we found and we proved that neutrophils are able to produce collagens in the subepidermal region, we wondered the relevance of this process. So we depleted the neutrophils using the IDTR model, and we investigated the skin using scanning electron microscopy. As you can see here in the control, collagen fibers are quite thick and compact, creating a rigid structure, while when you eliminate neutrophils, the fibers become less thick and more dispersed. So we saw loss of the rigid ECM structure when we eliminate neutrophils. We wonder whether these were also reflected on a change in the mechanical properties of the skin. So we used this custom-built tensile tensor, which basically allowed us to stretch the skin and to measure the stiffness of this tissue. And we found that when you eliminate neutrophil, the skin becomes less stiff. Then we wonder what was the program that was guiding this ECM production in neutrophil. And making a long story short, we identified that TGF-beta pathway is involved in this process because by blocking the TGF-beta pathway, barrier neutrophils are not able to produce ECM transcript. So we generated the MRP8-3 TGF-beta receptor 2 flox mice in which only neutrophils are insensitive to TGF-beta signaling. First of all, we saw that barrier tissue neutrophils could not produce ECM components. And we saw that when neutrophils are not able to produce this ECM, the skin again becomes less, the fibers become less thick, the diameter is increased, the skin becomes less stiff, and also the permeability of the tissue increases. As you can see here, when we put Evans blue in the neutrophil-specific TGF-beta knockout receptor mice, we see that the skin becomes more permeable, probably because in these mice, the skin architecture and ECM structure is affected. Last, we decided to see whether this program, this ECM production by neutrophils, were also relevant in a wound context. So we used a micro wound model using a 30G syringe tip, and we checked after 3 days. We discovered that a formation of an ECM ring around the wound, as you can see here, which is named for collagen 3. But this ring was not only formed by collagen 3, but also by other ECM proteins, such as collagen 101, laminin, elastin, and COL4. But most importantly, the formation of this ring was neutrophil dependent, as when we eliminate neutrophils or we impede neutrophil to produce extracellular matrix, as you can see here, the ring is not formed anymore. Then we wonder what could be the function of this ring. Since the localization of this ring was around the wound, we hypothesized that it was sort of a containment ring. So we used 2 kinds of permeability assay, one with Evans blue and one with a bacteria, Staphylococcus aureus. And in both cases, you see that in the control, both the Evans blue and the bacteria were not able to penetrate into the sterile tissue. Unlike in the neutropenic mice or the neutrophil-specific TGF-beta receptor knockout mice, where no ring is present, as you can see here, both the Evans blue and the bacteria were able to penetrate more deeply into the tissue. So as a brief summary, I showed you that neutrophils in the naive skin are able to produce collagen and actively deposit this collagen in the subepidermal area of the skin. And this confers more stability to the ECM and more rigidity to the skin. And this program is enhanced even in a wound context in which neutrophils surround the wound and they deposit ECM components to form this containment structure around the wound, which impedes microbes and foreign molecules to enter the tissue. This whole program is TGF-beta receptor dependent, because when neutrophils are not able to sense TGF-beta, they're not able to produce the ECM, resulting in less stiff skin, more permeable, and in the context of the wound, bacteria and molecules more easily enter inside the wound. So I would like to thank my PI, Andres Hidalgo, and all the members of the lab, also the collaborators and the funding. Thank you so much. And all of you for the attention.
Thank you very much, Tommaso, for this outstanding presentation. The two papers are now open for questions, so I remind you that you can write your questions into the chat part. Maybe I can start with the first question to Tommaso. I mean, it's very exciting results, and of course, all the work has been done in mice, and then you are, wondering whether you had the chance to do a few experiments in humans and see whether, you have similar profile in human neutrophils.
We did. We checked in human skin samples. We sorted the neutrophils out of human skin samples, and we saw that even in humans, neutrophils are able to produce collagens. And we also checked this in other barrier tissues, such as the lung and the intestine. So yes, these findings that I did in the mouse are actually translatable to humans.
Thank you. It's very, very exciting. Any other questions for the presenters?
I think we have one in the F&A, Sandrine. Maybe we do that now. That's beautiful work. Congratulations. And basically, it's about an interaction or correlation of the ECM production by neutrophils and fibroblasts. Do you have an idea, Tommaso?
Since we saw that actually the distribution of these two, of the collagen produced by fibroblasts and neutrophils are different, we actually saw that when we deplete neutrophils, ECM production by fibroblasts is not affected at all. So we think that these two kind of cells are not directly interacting to each other. They have specific role. They both deposit collagen, but in different type of region in the skin. So the fact that we see is that these two collagens, they're both required to the rigidity of the skin, but we found that the neutrophil one is subepidermal, and it allows the formation of super large fibers in the subepidermis, which is a really small region, but as I showed you, it has profound effect when it's depleted.
If you allow me, Sandrine, I would like to ask a question to Catherine. Catherine, can you hear me?
Yes.
Thank you very much. Really beautiful work. I have two questions. The first question relates to the B cells. You have beautifully shown that the plasma cells are polyclonal. What's your estimation? Is this a primary or a secondary event? Is it an epiphenomenon, or do you think it's causative?
You mean the B cell or plasma cell?
The plasma cell and antibody production, is it an epiphenomenon in the pathogenesis of HS? Or do you think it's involved in
For
early
HS pathogenesis?
Yes.
We are still working on this, but for now, I feel this is quite an early event. Because we see in perilesional skin that without any immune cell infiltration, there are epidermal abnormality. I think this disease is coming from keratinocytes, but we're still working on it. So
yeah, highly interesting. Thank you. Because it's relevant because we have BTK inhibitors and others, right, that target B cells. So we will realize quite soon. And the second question, if I may, beautifully shown the production of chemokines and fibroblasts here, the tertiary lymphoid structures, and TNF inhibitors. Do you know about IL-17 inhibitors? Are they able to destroy these formations?
I think we tried anti-IL-17. It doesn't work as well as anti-TNF or anti-CXL13. But I'm sure anti-IL-17 works in other ways, like, for example, to stop keratinocyte proliferation and like suppress T cells. Yeah.
Yeah. And if I may, Sandrine, a very last question, Tommaso, do you think what you have shown is beautiful? We know that under certain conditions, neutrophils just blast, right? And they may then release collagen. Do you think that may be relevant in the context of Catherine's work? So in fibrotic diseases such as HS, in a negative way?
Yeah, we actually, the next line of investigation, since we found that this process is occurring at steady state and in this bone-specific model, we're also wondering whether in fibrosis or in cancer, in other contexts, when collagen deposition is an active process, where neutrophils are in this context when they're actively depositing. So yeah, we're working on this, but yeah, really interesting.
Thank you very much. I think there is only one question left in the chat to Tommaso. The person is asking the level of expression when you compare neutrophils and traditional ECM producers. So is it the same or less or more?
Yeah, that's a great question. We actually compared RNA levels. Of course, fibroblasts have more transcript, but when we saw the immunofluorescence collagen production, we saw that overall, a fibroblast is bigger, so it has more collagen, it produces more collagen. But by area, since neutrophils are so smaller, neutrophils actually have more collagen than skin neutrophil, of course, than fibroblast. So that it's not comparable because of the size, but as well, what we think, because one question that we asked ourselves, like why neutrophils should produce collagens if they're already fibroblasts, which, they are the perfect cells to produce it. We saw that actually fibroblasts are really quite, they're not motile. They stay in their place, they don't not really move. But neutrophils are highly motive. They're highly motile. They move around the skin. And we figure out, since the subepidermal, in which we see the neutrophil deposit collagen, is a highly enriched collagen zone, all right, region of the skin, probably fibroblasts it's not easy for them to arrive to this region and to deposit collagen there. That's why I think with evolution, neutrophil took up this specific role because they're more motile, they're able to arrive in this highly ECM-rich area, and then they release collagen. So that's why we think it's not comparable, the amount, but the location is different, and was the key here.
Okay, thank you very much, Tommaso and Catherine, for these fantastic presentations. Now we can just start to close slowly this episode. Before we totally close, I would like just to remind you that there will be another ESDR Kitchen Sweet Sour episode on the 9th of July, presented by Christopher Elbrecht and Ricardo Grishaber-Bouye on CAR T cells and autoimmunity. Also, we would like to remind you that there will be an ESDR kitchen. I mean that all ESDR kitchen episodes and webcasts are available on the website of the ESDR kitchen. So you can look at them freely anytime. And to close this, I would like to remind you an important dates. the ESTR meeting that will take place this year in Anvers, Belgium, from the 10th to the 13th of September. And there will be a late-breaking abstract submission from the 1st to the 18th of July. And you can register as soon as the 9th of July. I mean, the deadline will be the 9th of July 2025. So thank you very much to all of you, and then we see you next time.